Pitt Hopkins syndrome is an autism spectrum disorder characterized by severe intellectual disability, extensive delays in development, distinct facial characteristics, irregular breathing, and has been linked with schizophrenia and epilepsy in patients. It is highly underdiagnosed and is often misdiagnosed as Rett or Angelman syndrome, and the FDA currently classifies it as an ultra-orphan disease, meaning that it's very rare. However, this probably reflects misdiagnosing. So, what causes Pitt Hopkins syndrome? Pitt Hopkins syndrome is caused by a mutation in TCF4. TCF4 is a transcription factor, and transcription factors work together to regulate gene expression, just as the components of a recipe combine to create a result. For cells, a good balance of transcription factors, all functioning properly, yields a healthy organism. However, if any one of these components is out of balance, such as if one adds too much vanilla, then you may have a bad result. Such is the case with TCF4 and Pitt Hopkins syndrome. There are no specific therapies available. Patients may access treatment for specific symptoms such as hyperventilation, epilepsy, and constipation, and they may engage with physical, occupational, or speech therapies. However, more research needs to be done to understand TCF4, its function, its structure, and how it results in the symptoms exhibited by Pitt Hopkins syndrome patients. Now, let's learn a little bit more. Typically, proteins need a specific structure to perform a specific function. However, with intrinsically disordered proteins, they can perform a function while having several rapidly interconverting conformations. They're an emerging field of research and represent about 30% of proteins present in bodies. They're critical to TCF4, as TCF4 is an intrinsically disordered protein.
Okay, now let's look at how researchers use simulations to study proteins and to study how proteins interact with each other. Study their function, what they look like, how they interact. Very cool. To do so, let's access the Wake Forest supercomputing cluster. It's called the DEEP cluster. Here is essentially the homepage, and now let's go ahead and find our directory that has all of our simulations. These simulations were ran as part of an academic course taught by Sam Cho at Wake Forest University. Here are all the files that you need to run a simulation. There are quite a few. But the really interesting thing is what they actually look like, so let's go ahead and take a look at that. Here's our protein as it exists in solution. And I say in solution because when we study proteins, we represent them as being in water. Why do we do this? Well, humans are largely water. So our proteins, our DNA, our cells, they're all essentially just swimming around in a bunch of water. What we have here are all the little water molecules. This makes everything really confusing, so let's just get rid of those. Now what's an MD simulation? An MD simulation predicts the interaction energies between nearby atoms and then says, okay, this is how much they repel each other, this is how much they attract each other. Over a certain time period, how much are they going to move? Okay, cool. Pause, recalculate the interaction energies, and let's run again. It does this for every single atom in our protein. And it's very computationally expensive, which is why you need to have a supercomputing cluster. Now, these trajectories are on the order of nanoseconds, which is not a very long time for most things that we do, but as far as proteins are concerned, it's long enough to get a good sense of what's going on. So let's go ahead and look at what these simulations actually look like. This is a mini movie of 1 nanosecond. It's the same molecule, all we've done is change how it's drawn. We've changed the background color, and we've isolated part of it to highlight it. Whoops. Here we go. It's a lot of jiggling. How can we make sense of this? Well, what you do is you tell the computer, okay, let's take two atoms. What's the angle between them? How far apart are they? What's going on? Analyze the entire protein over the entire trajectory and see if we can find any patterns. Through this method, we can gain a better understanding of the structure and hopefully the function of proteins, and in our case, the intrinsically disordered proteins.